This video lecture is over Chapter 2, Mitosis and Meiosis. Most of this material should be reviewed from Biology 112, Cell and Molecular Biology. And as such, I'm going to move pretty quickly through this material. I've condensed this lecture down significantly. And please refer to the study guide to kind of see what material I'm expecting you to know for the test in which I'm not going to be testing over. So this chapter begins by talking about how cell structure is really closely tied to genetic function. And there's a good overview of cells. So if you've forgotten the difference between a prokaryotic and a eukaryotic cell, please go back and look at that. If you don't know what some of the common shared features of cells are, that all cells, whether they're prokaryotic or eukaryotic, have plasma membranes, DNA, and ribosomes. Make sure you go back and look at that. Additionally, I expect you guys to be familiar with the organelles found in a eukaryotic cell. I could potentially ask or test over these on your exam. So know the general functions of the organelles. Today, I'm going to just speak about the centriole and the nucleus, since that's going to be most pertinent to what we're talking about today. So the centriole is an organelle, and it's found in a region of the cell that we call the centrosome. In both plant and animal cells, they have these centrosomes, and it helps organize the spindle fibers, which are microfilaments. Those are proteins that kind of act like a railroad line, like a transport line in the cell for the movement of chromosomes during either meiosis or mitosis. All cells that contain DNA in eukaryotes, they are formed or compacted into what we call a chromosome. And chromosomes exist in most organisms in what we call homologous pairs. So that means we have 2 copies of each chromosome. In humans, we have 46 chromosomes. Okay, those are comprised of 23 homologous pairs. So we have 23 different chromosomes from the mother and the father. And our diploid number, so the total number of chromosomes, would be 2 to the n, where n is the number of chromosome pairs. So 2 times 23 equals 46. So we have 46 chromosomes in each cell. Now, homologous chromosomes, they are similar to each other, but they are not identical. So when I teach this course in person, at this point I would normally say, okay, turn to the person next to you. Look at them, look at some of their traits, look at the person on the other side of you. Do you contain the same genes as the people sitting next to you? And then I ask for a show of hands. How many of you think you have the exact same genes? Or this person next to you perhaps has a gene that you don't have. And surprisingly, a lot of students fail to recognize that they have the exact same genes as every other human on the planet. Unless something went drastically wrong during fertilization and embryonic development, you have all of the same genes as the person next to you. Now, the difference when you look at the person next to you and they have brown hair and you have blonde hair, or they have blue eyes, you have brown eyes, is the fact that even though we contain all the same genes, within those genes, there can be point mutations that alter the function of the proteins that those genes encode for. And those proteins then give us the variation when they're slightly altered. That's the variation that we see. Now, that doesn't mean you have a different gene. What that means is that you have a different allele of that gene. And we'll talk more about alleles and genes in our next chapter on Mendelian genetics. But for right now, just remember that a homologous chromosome, if we're talking about chromosome 21 in humans, right? You have a chromosome 21 from your mom, chromosome 21 from your dad. They have all the same genes on those chromosomes, but there may be some differences in the genetic sequence of those genes that cause variation. Our chromosomes also contain a region that is constricted and condensed, and that is called the centromere. Okay, the centromere is the location in which, when we undergo either mitosis or meiosis and we replicate our DNA and we form our tetrads, this is where the two chromosomes, what we now call the sister chromatids, would attach to each other, and that the region is the centromere. Now, depending on where that centromere is found in relation to the length of the chromosome, we can designate that chromosome. If it's found directly in the middle, we would call that chromosome metacentric. If it's a little off, it's somewhere between the middle and the end, then it's called submetacentric. And at this point, you'll notice we have two different lengths of our arms of our chromosome. So here they'd be the same, so we can't really designate which is which. But as soon as we are able to designate, because one is smaller and one is longer, the smaller one is always designated the P arm, and the longer arm is designated Q. You can remember this because if we're talking about increasing in size, P comes before Q, so P is smaller than Q. As we move closer to the end, not all the way at the end, but closer to the end, so you can see here's our chromosome, there's the centromere, it's close to the end. This is what we call acrocentric. Okay, again, it's really difficult to designate a P and Q, even though technically this would be the P. Finally, if it's all the way at the end, that chromosome would be designated telocentric. So make sure you understand the difference between these. The next section of this chapter talks about our cell cycle. So the cell cycle are the stages of normal cell functioning that a cell goes through. The one that we're going to spend the most time talking about today is mitosis, in which we partition our chromosomes. We then take those replicated chromosomes and divide them into two daughter cells that are diploid. Mitosis has two parts to it. It has the division of genetic material, that's what we call karyokinesis, and the division of the cytoplasm and the cell itself, which is cytokinesis. The cell cycle itself then can be divided into that stages of mitosis, in which we are dividing the genetic material, and the stage at which we're preparing for that, which is what we call interphase. So when a cell is in interphase, it is going about its normal cell business. It is making proteins. It's engaging in cellular functions, cellular maintenance. Okay, we can divide interphase into three main parts. We have what we call gap 1 stage or phase, gap 2 phase, and between these we have the S phase. Now during gap 1, what the cell is doing is it is essentially growing and getting ready to be able to divide. When we are in S phase, the cell is synthesizing additional copies of DNA so that when it divides, it'll have a copy for each cell. In gap 2, it then is preparing the DNA and making sure that synthesis, that all the DNA is good, in good condition to be able to divide. You'll notice there's also this gap 0 phase. Some cells that don't divide, let's say we take a neuron. Okay, that's neuron, once it's developed, it's really not going to divide anymore. It's not replicating into others, or for the most part, it's not going to divide anymore. So it goes into a stage in which it just stays in gap 0 and it's just maintaining normal cell functions. It's not trying to grow anymore. It's not trying to get bigger so it can divide. It just stays in that stage. Now, what dictates when we move from gap 1 into S, or from S into gap 2, or even from gap 2 into mitosis, there are cellular checkpoints in which the cell is monitoring the condition of itself and deciding whether it goes in to S phase or mitosis. These checkpoints are monitored by proteins called cyclins and kinases. Okay, these are proteins that are utilized in a lot of biochemical pathways and that are necessary for the cell to then engage in DNA synthesis, and DNA replication, and DNA repair. And so cyclins are kind of, we kind of think of them as governing protein, governing cell cycle proteins. That's why we named them cyclins. They are integral to the cell cycle. Okay, and the kinases are the enzymes that activate these cyclins. So at these particular checkpoints, so at G1 S checkpoint of what decides if we can move from G1 into S phase? Well, the cell has been monitoring its size based off of those cyclins, the amount of cyclins that are being produced, what type of cyclins, and so that can trigger, once we have enough of those, it can trigger us to move into S phase. Okay, it's also evaluating the condition of the DNA. Is the DNA suitable to be replicated? There's cyclins that get produced to help repair DNA. And if we have a lot of those, then we might not be moving into S phase. Now, the next checkpoint I want to talk to is when we move from G2 into mitosis. And at this point, what the cell is monitoring is, did we replicate our DNA? If DNA replication is incomplete, then it's not going to move through. And if the DNA is damaged, it's not going to move through. And again, those cyclins can help tell the cell, based off if it's producing a bunch of repair cyclins, hey, maybe we're not ready to move into mitosis. The last checkpoint would be the M checkpoint, in which we move through mitosis and start beginning that actual mitosis. And that's going to be really dictated whether we form our spindle fiber system to attach to our centromeres and help pull those sister chromatids apart. Mitosis itself has 5 discrete stages. Those stages are prophase, prometaphase, metaphase, anaphase, and telophase. Okay, you're responsible for knowing and being able to identify those stages from images, like what you see here. So we'll go through those real quick. If you're having a tough time remembering these, I would suggest making a mnemonic in which you use the first letter of each of these cycles to help remember it. I like mnemonics that are a little bit weird or perhaps a little bit shocking. They tend to stick in my head a little better. The one that I use to remember this is punching puppies makes anyone terrible. Right? That's what I write to. Punching puppies makes anyone terrible. Let's go through these stages and talk about what are the discrete things that are going on. So prophase. During prophase, we have our sister chromatids. Okay, so a sister chromatid is a identical copy of the chromosome that was made during S phase. So it's completely identical in that it has the exact same alleles, right? So what we see happening in the cell during this stage is that our centrioles, they divide and they start moving towards the 2 poles of our cell. They're going to establish those poles. Additionally, what defines prophase is that we start to see the nuclear envelope break down, but we can still see it in the cell. So here we can still see the nuclear envelope. We're seeing the chromosomes start to condense and become visible. So our chromatin is condensing and becoming visible. And our sister chromatids are going to start to connect at their centromeres. So our next stage is prometaphase. And prometaphase, what defines this stage is that the nuclear envelope is gone. So we no longer see the nuclear envelope at all. Additionally, our sister chromatids have combined at the centromere, and so we have our chromosomes moving in pairs towards the middle. Our centrioles have reached their poles, and then the other thing that we'll notice, that you didn't see in prophase is that the spindle fibers are now attached and starting to form. Okay, so they're attaching to the kinetochores, to those centromeres, and they're starting to position those chromosomes for division. That brings us to metaphase. So in metaphase, we now have aligned all of our sister chromatids on the metaphase plate in the middle of the cell. All of the spindle fibers have attached to the kinetochore. And what's the kinetochore? So if we look at our centrosome, okay, so here's the centromere. All of the centromeres have aligned on that metaphase plate, and the kinetochore are the proteins that are associated with the centromere. And this is where our spindle fibers bind and help pull our two sister chromatids apart. What those proteins are that we are associated. So we have the kinetochore proteins, and then there's a couple other proteins that are important. We have cohesion, which are these proteins that actually hold the sister chromatids together. They kind of form between the two strands. Okay, we have sugation, which is a protein that is found primarily in the centromere, and it protects the cohesion proteins from being degraded. Okay, so cohesion exists all throughout. Sugation is found primarily at the centromere. Okay, you can have a little bit in other areas, but primarily this is where we find it, and it helps kind of protect it. Now, sugation is a Japanese term. It kind of refers to armor. So again, it's kind of this armor that's protecting the sugation. And what we see occurring around metaphase is we start getting the production of an enzyme protein called separase. And you have to love the simplicity of who named these, at least these two proteins, cohesion and separase, because the function of separase is to degrade cohesion. The function of cohesion is to keep the sister chromatids together. And so what happens is, as we start into metaphase, or as we start into, mitosis, we start producing separase. And so that separase starts breaking down the cohesion. The cohesion that's going to break down first is the stuff that's not associated with the centromere. So this will be gone well before we ever align on the metaphase plate. This takes a little bit longer. But by the time we get to the metaphase plate, that separase starts to break down and separate, and this cohesion, right? The sugation can no longer kind of hold it together, and it starts to break apart. In anaphase, we have what's known as disjunction. So disjunction is where we have, in this case, sister chromatids separating. Okay, that is a disjunction event. We're being pulled to the two poles, and we migrate to those poles. At this point, these sister chromatids, since they've been separated, are now called daughter chromosomes. Okay, so the daughter chromosomes are moving to the opposite poles via disjunction. And then the last phase of mitosis is telophase, in which the nuclear envelope starts to reform. Our spindle fibers start to disappear. And we also start seeing cytokinesis form. So we start getting the pinching of the cellular membrane, and cytokinesis will then take place. If we're in a plant cell, we'll start getting the formation of the cell plate. Those are the defining characteristics of telophase. So please make sure you understand these. I know these should be reviewed. I probably could have gone through them much quicker for all of you, but if you had forgotten, make sure you understand all of those stages. Now, what's a little bit more complex is the process of meiosis. So meiosis is a process in which we reduce the genetic content found in our daughter cells down to where we're just haploid, meaning we only have one copy of all the chromosomes. So instead of having 46 chromosomes in a human, we would have 23. And we use meiosis for sexual reproduction. So our gametic cells, those are our sex cells. The sex cells would go through meiosis. And in order for us to go from a cell that then doubles its DNA during synthesis, so it makes a copy of everything, and then divides into haploid cells, we need to go through basically 2 stages of mitosis. Okay, at the end, we're going to reduce our genetic material by half, and we're going to produce what we call haploid gametes or spores that contain only one member of a homologous chromosome pair. Those 2 stages we term meiosis 1 and meiosis 2. So meiosis 1 is the reductional division, in which we are reducing the amount of genetic material by half. And meiosis 2 is then where we do the equational division, where we make sure that then those cells only have one copy of half of the chromosomes. That'll make a little bit more sense when we go through this. Okay? Again, just to remind you, DNA synthesis occurs during interphase, during the S phase, and it's well before the beginning of meiosis 1. It won't occur again before meiosis 2. We don't do any more DNA synthesis. This is a figure taken from your book that shows the difference between mitosis and meiosis. It's a really great figure because a lot of students get confused at when do we become haploid in the cell? When do we lose half of this? And it's confusing because you tend to focus on number of chromosomes and not which chromosomes they are. So I'm going to go through this and try to outline to show you that at the end of meiosis 1, we have actually lost half of our genetic material. So let's take a cell that is diploid, and it has 4 chromosomes. So that means there's 2 homologous pairs. In this case, we have one that is a large chromosome and then another that is a small chromosome. And we'll use the blue and orange designation to kind of be father-mother. So we'll say the blue is the father, the orange is from the mother. So we have this chromosome 1, which is the long chromosome, and chromosome 2, which is the shorter chromosome. So during S phase, we are going to make copies of all of these chromosomes. Okay? Those copies then, during mitosis, right, they would form sister chromatids and combine at the centrosome. The same thing happens in meiosis 1. Okay? At the end of mitosis, right, those sister chromatids get pulled apart to where you'd have 2 daughter cells, and again, they're going to have a long blue chromosome from the father, so the chromosome 1 from the father, chromosome 2 from the father, chromosome 1 from the mother, chromosome 2 from the mother. Okay, so they are still diploid. However, at the end of meiosis 1, okay, what happens in meiosis 1 is the chromosomes align, but then they form these what we call tetrads, in which both of the chromosomes 1. So you see here is the mother's chromosome 1 that's made the two copies, the sister chromatids. Here's the father. They form a tetrad in which they combine to each other. They hook. There's a chiasma that forms. And then when they get pulled apart in meiosis 1, you'll see that the tetrads get separated. Okay? So the homologous chromosomes get pulled to the end. So now we have, in this cell, we have 2 copies of the mother's chromosome 1, but we have no copies of the father's. The father's chromosome 1, both the copies are in this cell. Likewise, here we have 2 copies of the father's chromosome 2, no copies of the mother's. The mother's over here. So these cells are, at this point, haploid. Okay? Even though you may count and say, oh, this is 1, 2, 3, 4, 1, 2, 3, 4, and here we have 1, 2, 3, 4. Why, Dr. Smith, why aren't these diploid? No, these are the exact same chromosome for all intents and purposes. So we have the mother's 1 and the father's 2. We do not, here we have the mother's 1, the father's 2, the mother's 1, the father's 1, the mother's 2, the father's 2. We don't have that here. We've lost half of our chromosomes. They're gone. Yes, we have 2 copies of the same chromosome, but that doesn't matter. We've lost half our genetic material. Now, we have a second meiosis division, which we call meiosis 2. This is now the equational division, in which we want to make sure that we only have a single copy of each of those chromosomes in there. So we divide again, and this would be a division very similar to mitosis, in which the sister chromatids this time get pulled apart, not the homologous chromosome pairs. And now we end up with 4 gametes, and there's a single copy of the mother 1, single copy of the father 2, single copy of the mother 1, father 2, single copy of the father 1, mother 2, single copy of the father 1, mother 2. So hopefully that outlines how we go in the reductional phase to haploid, and then in the equational division, we just have a single copy of the chromatids, not 2 chromatids. Something else that I want you guys to be aware of, there are some additional stages in meiosis. So we have the same, prophase, metaphase, anaphase, telophase. Okay, we designate between these 2 divisions as prophase 1, metaphase 1, anaphase 1, telophase 1, and prophase 2, metaphase 2, anaphase 2, telophase 2. We're not going to be concerned with prometaphase for meiosis. Why? Because in prophase, there's a series of things that happen during prophase 1 that are really important, and so they we tend to focus on those rather than where prometaphase maybe actually be found. I do want you to know these 5 stages of prophase 1. They are leptonema, zygonema, pachynemia, diplonema, or dikinesis. Sometimes you'll see these called leptotene, zygotene, pachytene, diplotene, dikinesis. How you can remember these, again, use a mnemonic. I don't care what mnemonic you use. It's LZPDD. I like to use kind of wacky, weird ones. Help me remember it. The one that I remember for this is for leptonema. So L, large, zits, pack, dripping, discharge. Okay, yes, I know that's gross, but that's what I remember. Large, zits, pack, dripping, discharge. If that one's too graphic for you, come up with your own. But that's how I remember it. Okay, what happens during these? Okay, well, during leptonema, our chromosomes are starting to orient themselves. During zygonema, what we find is that we now have what we call bivalence, in which our chromosomes have formed next to each other. They're still very tightly bound to each other, so the cohesion is keeping them close together, and so we don't really see the tetrad. When we can actually see the 2 tetrads, that's what designates pachynemia. Okay, so here we can see that the tetrads have formed. Diplonema, what distinguishes diplonema is now we see the crossing over of the father and the mother tetrads, and that crossover point is where we exchange genetic material. So we have genetic exchange between it. You can see that here, right? So what happens is, because these have roughly they have the exact same genes, there's areas on the chromosome that are likely in which you can have adhesion of one chromosome to another chromosome when we're starting to break apart some of those cohesion molecules. And when that occurs, we have enzymes that come through that try to fix the DNA because it's stuck together. And so they snip the DNA. And in snipping it and religating it, what sometimes happens is that we have a part of the father's chromosome gets put on the mother's chromosome. And this is a really elegant situation in which now we can have additional DNA diversity during meiosis. So it's not just the random assortment of our chromosomes, but here we also have the shuffling effect that can occur by having crossover. Okay, so that happens during diplonemia, and in diakinesis, that crossover, we have what we call terminalization, and it kind of, that's where we're actually doing the cutting, and the stuff gets exchanged. Okay, so that's prophase 1. In metaphase 1, what happens? Well, our homologous chromosome pairs have now aligned on the metaphase plate. Okay, so we have our tetrads, and the homologous pairs then in anaphase 1, which gets separated, what moves to the poles? Homologous chromosome pairs. Okay, telophase, those reach the poles, we start making the nucleus, and we have cytokinesis. Then we go through another round of division. So prophase 2, okay, in which again, we're now going to be moving. This is exactly like mitosis at this point. We're going to move our chromosomes, attach the spindle fibers, start aligning them towards the metaphase plate. Metaphase 2, we've aligned on the metaphase plate. And then we have disjunction. So again, there was disjunction here. We have disjunction here in anaphase 2, in which now sister chromatids are moving towards the poles. So anaphase 1 disjunction is of homologous chromosomes. Anaphase 2, the disjunction is of sister chromatids. Now, you're going to be asked a question in your study guide. There's a question about this disjunction, in which you should probably kind of draw it out to answer some questions. It asks about what happens if you have non-disjunction. So non-disjunction would be a failure of these homologous chromosomes to separate from each other. So if we had non-disjunction in anaphase 1, what would happen, instead of having these two separate, non-disjunction would end up with, say, perhaps this whole body moving into the one cell. So we'd have down here a cell that has both of the large chromosomes, right? And then one of the small one. And then the other cell up on top would only have its small one, because we had a non-disjunction event with these two separating. They didn't separate normal. This one stayed with that guy and went down. Okay? That would result here. So there's a question in which you have to draw out the non-disjunction to kind of figure out what would happen. What would you be left with? Last, let's kind of summarize everything. What's the point of meiosis? Why have this double division? And the beauty of eukaryotic cells and then their ability to undergo meiosis when they decide to, is that it allows for an increase of genetic variation between generations. So if we just had mitosis, we're essentially cloning cells. We have a clone, there's not any new variation that gets added into it. When we go through meiosis, we now create a cell that has half of the DNA, and it's gone through the shuffling process, and we now can then have fertilization. So we can take a gamete from a mother, gamete from the father. Those two cells can fuse, have the appropriate number of chromosomes, and then start to develop. And they're going to have different alleles than, say, the mother and the father individually had, because there's this idea of kind of shuffling the deck and getting a different amount of genetic variation. We'll talk all about that with Mendelian genetics of what we can get and how we can start to predict what are the probabilities of receiving a particular trait through going through this process of genetic fertilization and recombination that's associated with meiosis and fertilization of two haploid gametes.